Okay, and I think we are live. Hello, everyone, and thank you for coming to our session today as part of an ongoing series this year, hosted by the Foundation for Food and Agriculture Research in celebration of our 10-year anniversary. Today, we have four fantastic speakers who will share both innovations and challenges related to plant health. My name is Ryan Kamela, and I'm the Scientific Program Officer supporting the crops portfolio at FFAR, and I'll be your tour guide today. If you aren't familiar with FFAR, we were created by the 2014 Farm Bill to build public-private partnerships that fund bold food and agriculture research. Over the past decade, we've awarded more than 360 grants, amounting to over $700 million in funding. What's more, we've worked with over 550 partners across the food and agriculture value chain to do this. Lastly, FFAR matches every federal dollar with a dollar from a non-federal source, and today that match ratio stands at 1 to 1.4. As we celebrate 10 years of FFAR, we are looking back to our decade of impact and showcasing a vision for the future. Throughout the year, we're hosting monthly webinars just like this to feature new areas of innovation. And you can see our previously recorded webinars and sign up for future sessions through our website, foundationfar.org/impact. And this, of course, would not be possible without our fantastic grantees, partners, and collaborators. Thank you to all. We are forever grateful for your support. Lastly, please feel free to submit any questions to the chat box, and we'll be sure to answer them during our final Q&A segment later towards the end. And without further ado, I'd like to introduce our first speaker, Dr. Jan Leach, for the segment titled What is Plant Health and Challenges Posed by a Changing Climate? Jan is a plant pathologist who studies the molecular basis of plant disease susceptibility and resistance, and how these responses are influenced by interactions within the phytobiome. She is a University Distinguished Professor in the Department of Agricultural Biology at Colorado State University, and is also a past president of the American Phytopathological Society and the International Society for Plant Pathology. Jan is also a member of the U.S. National Academy of Sciences. Welcome, Jan.
Thank you. Thank you so much, Ryan. Well, first, I want to congratulate FFAR on their 10th anniversary, and thank you for the opportunity to present some current insights into how climate change is predicted to or is actually already affecting plant health, and some ideas and approaches that are underway to help mitigate it. Climate change, as you are well aware, is already dramatically impacting crops across the globe. And the predictions are that for many plant diseases and pest interactions, it's going to continue to worsen. The example on this slide is black sigatoka disease of banana. So since the 1960s, the infection risk for this particular disease has increased by around 45% across banana growing regions in Latin America and the Caribbean. And this is because climate change has increased canopy wetness and temperature conditions that are such that they're much more conducive to pathogen infection. So we know already we're seeing the impacts of climate change in many cases. But even if we don't consider climate change, if I can have the next slide, please, we know that the global burden of pathogens and pests is huge. It already ranges from 10 to 30%, depending on the crop species around the world. Unfortunately, these losses vary with region, and the worst impact is on those regions which are food deficit regions and that have fast-growing populations. We've also found that these incidences of disease and yield loss caused by diseases and pests are frequently associated with emerging or re-emerging diseases and pests. If I can have the next slide, please. We need to understand the impacts of climate-driven pathogen and pest migration on agricultural health. With shifts in environmental climates, pathogens and pests are invading new ecosystems at unprecedented rates. This particular slide shows the global incidence or emergence of new crop diseases over a 7-year time frame. And interestingly, in these new encounters, the plants are often more susceptible to the pathogen, possibly because they've not co-evolved. So the plants do not have any immunity to those pathogens or pests. A particularly frightening example comes from China, which is an important producer of several major crops. The relative area of cropland affected in China by damaging pests and diseases has quadrupled in the past half century, and it could easily double again by the end of the 21st century. If I can have the next slide, please. So we need a deeper understanding of the dynamics of biotic and abiotic interactions and how they impact agriculture, biotic being pathogens and pests, abiotic being climate and imposed stresses such as high temperatures or drought or cold. Work over the past decade has shown for many crop species, climate change imposed stresses exacerbate existing problems caused by pathogens, pests, and weeds. We've shown, for example, that rice is more susceptible to bacterial blight pathogen at elevated temperatures. And these temperatures are well within the ranges that we're already seeing in rice producing areas around the world. If I can have the next slide, please. We need a deeper understanding of the dynamics of biotic and abiotic interactions and how they'll impact agriculture. Work over the past, I'm sorry, I wanted to move on to this thought here that in a global analysis, diseases will counteract predicted crop yields that we see under the climate change conditions. So you've heard in the reports from ICPP that we're seeing in some cases, as the climate increases, and we'll talk a little bit more about this in temperatures and humidity, that we're seeing increased disease. I mean, we're seeing increased yields in these areas, and you can see the green arrows pointing to rice increased yields at certain latitudes. But these are going to be counteracted by increased risks for pathogens and diseases. So the climate change, while we may see better yields in some instances, often that's going to be associated with larger yield losses from pathogens. I can have the next slide, please. What's really frightening is that the climate-imposed stresses are negating our efforts to reduce the impacts of pathogens and pests. So looking back to our example of bacterial blight of rice, resistance genes that have taken breeders years to identify and introduce into rice are often rendered ineffective at high environmental temperatures. In this case, the FFAR gene, which is one of the most widely used R genes throughout the rice-producing areas of Asia, is almost completely ineffective at high environmental temperatures that we're already seeing in parts of Asia. On the other hand, we are also seeing that plants bred to tolerate higher temperatures. So as the breeders introduce the genes to make the plants more resistant to high temperatures, we put them out in the field and we find that they're highly susceptible to pathogens. So clearly, we don't have the luxury of focusing on improving for one stress at a time. We really need to embrace the complexity, and our science needs to fundamentally shift to systems-based innovations and approaches that address complex combinations of these stresses. If I can have the next slide, please. So why haven't researchers embraced the complexity? Well, part of it is because we need to consider this as a system, not just one stress at a time. The studies have become more complicated. When do we apply the stresses? For example, if we're applying heat and pathogen, do we apply them simultaneously, staggered, or continuously? And we already know that the outcome of combined stresses frequently varies from what we would observe with single stresses. For example, I just presented where combined heat and pathogen stresses can render plants susceptible that would normally have been resistant. or they can render heat-tolerant plants diseased. So we have much to learn about how plants and pathogens interact in these scenarios with non-optimal climatic conditions, and we need to think about how to design these very complex studies. If I can have the next slide, please. So in the next few minutes, I'd like to propose some mitigation strategies as we look to the future. While I've argued that we need to move beyond studying one stress at a time, those previous studies are the foundations for what we need, and we need them to move forward. We've learned a great deal about environmental changes and how climate change will, and what it's going to throw at us in the future, and what kinds of stresses these are going to impose on the plants. So for example, depending on the location, we might expect heat stress or cold stress, or we might see increases in flooding or drought, both types of water stress. We often think of increased atmospheric carbon dioxide levels as associated with faster growth and more biomass in plants, but unfortunately, it's now being shown that these plants are less nutritious because they contain more starch and less proteins and fewer vitamins. We also know that under these conditions, the cell walls and other structural features are altered, and so these may render the plants more susceptible to pathogens or diseases. So for some pathogens on plants, higher temperatures is going to increase the production and delivery of virulence factors by the pathogens. Now, virulence factors are the proteins or compounds that enable a pathogen to invade a plant and cause disease. And what is alarming is that this increased virulence can sometimes enlarge the host range of the pathogen, enabling it to colonize plants or other causing more damage or at a higher or new hosts. Disease risk also will increase as pathogens and pests are introduced by winds or by humans into new areas with favorable climate conditions. And if the disease hasn't been present in these areas before, the crops are frequently more vulnerable, as I've mentioned before. On a similar note, as climate changes, we're talking about shifting crop production into new areas where the climate is more conducive for the crop. But again, unfortunately, these newly introduced crops are frequently more vulnerable to pathogens that they've not encountered before. We're also interestingly seeing the loss of efficacy of a plant's immune system due to climate change imposed temperatures or droughts or other conditions. And I'll talk a little bit more about that later. If I could have the next slide, please. So our mainstay in thinking about the future, how are we going to genetically alter plants in order to make them more resilient under climate stresses? Our mainstay now for helping plants thrive under the threats of disease or environmental stresses is to improve their genetics through breeding or newer technologies such as genome editing. But since we don't know much about how the disease and environmental stresses interact, we don't really have a clear idea of what genes or traits we need to pursue. We found, for example, that many R genes lose efficacy at high temperatures, and that a few will maintain function under elevated temperatures. Deciphering the molecular mechanisms of why these genes retain resistance function under less than favorable conditions may provide important clues for improving crop resiliency. Another strategy is to improve the downstream, if I can have the next slide, basal defense response gene activity. In plant immune responses, the plants use protein receptors to detect the presence of pathogens or pests and to activate expression of defense response genes. If I can have the next slide, please. I liken these receptors to generals who direct the immune responses. Defense response genes are the soldiers that carry out that activity. And if you don't have good soldiers, the generals can shout all they want, but no one's going to carry out the orders. So in studying these downstream defense response genes, we found that many are activated not only during disease resistance responses, but also some are activated during abiotic stresses, such as high temperatures or drought. I can have the next slide, please. We've accumulated these good alleles of DR genes into rice and have shown that the more active alleles of the genes we have, the more resistant the plants are to disease. And importantly, this resistance is long-lasting. For example, in this study, we showed that the variety with 5 good alleles stood up in the field at 2 locations for 11 years, or 22 cropping seasons for rice. If I can have the next slide, please. With further exploration, we demonstrated that the effective alleles were active because they contained particular organizations of cis-regulatory elements in their promoters, which we call cis-regulatory modules. The presence of these conserved PRIM, or cis-regulatory modules, is associated with gene activity and defense responses. If I can have the next slide, please. With funding from FFAR, we've shown that some of these CRIM are common across stress responses, in particular disease and heat tolerance. Our goal is to use these CRIM to help breeders develop markers that will enable simultaneous breeding for tolerance to multiple stresses. If I can have the next slide, please. I think I altered the slide here. Yeah, thank you. Although I've talked mainly about how pathogens and insect pests, I'd like to acknowledge that weeds are major pests whose negative impacts on agriculture will increase under climate change. But we can learn from these hardy weeds. A strategy that's being supported by FFAR is to exploit the amazing resiliency of weeds for traits to develop climate-resilient crop species. Next slide, please. I've talked a little bit in a previous slide about other strategies that we can use, such as understanding more about how the plant's microbiomes can help plants tolerate stresses, climate stresses, but we really don't know how they work in combined stresses. And we also need to think about moving plants into pristine environments and using regulatory phytosanitary requirements before they cross borders. And also looking at plant diversity and how we can use that as a reservoir to improve crops and keep this diversity moving forward. This slide shows a recently published National Climate Change Roadmap, where we used a horizon scan strategy and using over 60 participants to study a framework for research to ensure that agriculture remains resilient in the face of climate change. And with that, I'd like to thank you again on the last slide, and to again congratulate FFAR on their 10-year anniversary. Thank you.
Excellent. Well, thank you, Jen. Spectacular presentation. Next, I'd like to introduce Dr. Alonso Favela, who will walk us through this segment discussing water as an influence of plant health. Alonso serves as an assistant professor in plant sciences at the University of Arizona, specializing in plant microbe interactions. His work explores the complex connections between agricultural plants and various microbial communities, aiming to uncover their significant influence on ecosystem functions amidst the Anthropocene era. His ultimate goal is to pinpoint sustainable strategies for addressing contemporary agroecosystem challenges. Alonso, the floor is yours.
Good morning, everyone, and thank you, Ryan, for the presentation, for the introduction, and thank you for the invitation to speak with you all. Next slide. Can't see my, okay. I'm honored to be a part of this group of speakers, and I'm very excited to talk to you all today. Today, I'm going to be discussing water, as Ryan mentioned, and as someone who grew up in Phoenix, Arizona, and is now a faculty member at the University of Arizona, I'm acutely aware of the importance of water to our society and to our livelihood in this arid state. And currently, our water usage here is not sustainable and needs to change. And I hope to become a leader in incorporating how the microbiome can address some of these agricultural water issues. Next slide. So the availability of fresh water is rapidly changing worldwide, creating a precarious future that requires urgent attention from policymakers, the public, scientists, and farmers. This map shows the predicted change in Earth's water distribution, with red regions predicting a decrease in water storage, and blue regions predicting an increase. This image is generated using the NASA GRACE satellite. The satellite is actually really impressive. It measures the gravity changes on the Earth's surface caused by the presence of water. And the reason I'm showing you guys this figure is that it tells a clear story about how water scarcity is predicted to change across this planet. It shows that climate change will result in unequal distribution of water, with a predicted increase in water scarcity for about half of the world's population. Those that have water now are going to get more, and those that have less are going to get less. Next slide. And even though we know climate change is predicted to change the hydrology of our planet, this hasn't curbed how we're using water. Since 1901, we've steadily increased our water consumption across the planet. And as states and countries become more industrialized, the general trend is that they typically increase their per capita water usage. So there's a huge and growing demand across the planet for this water as our water sources are becoming even more erratic. Next slide. And nowhere in the United States is this issue clearer than in my home state of Arizona and in the Southwest as a whole. Here are a couple of recent headlines highlighting the tense situation over the Colorado River, which supplies water to 6 states in the Western United States. Many are starting to ring the alarm bells about our water limitation. A recent mega drought in the Southwest caused our water levels to dip to lower levels than it ever has, and predicted cuts have been planned for the residential water usage of the state. So people are starting to feel the pressure of their water usage at the residential level in their own homes. And while here I'm only highlighting the water limitations in the Southwest, in typically rain-fed regions, droughts and changes in hydrological patterns have resulted in losses in yield and productivity in those regions as well. So this is a big issue. Next slide. So you might be asking yourself, where is all of our water going? And it's basically going to the production of food, feed, and fiber. It's going into agriculture. Agriculture accounts for the largest loss of fresh water, with 80 to 90% of our total US water going into agriculture. To put that in perspective, that's about 5.5 amounts of water that is equivalent to the volume of 5.5 Lake Eries. So this is a huge amount of water that's going into agricultural systems. And even though they're very consumptive, irrigated lands provide 40% of the world's food for less than 20% of the cultivated area. They're a very useful source of food. And for example, in the United States, irrigated lands provide important food during our winter season. Without farming in Arizona and California, we wouldn't have vegetables, citrus, and salads during our cold months in the Midwest and East Coast. So irrigation is important for global food security. Next slide. And the reason irrigation is so important is that plants need water. Water is the most limiting abiotic factor to plant growth and productivity. Water is required by plants to carry out photosynthesis and transport nutrients throughout their body. And water flows from areas of high water potential to low water potential. In this figure, you can clearly see how that sort of works in practice. Water potential in soil is around 0, and then specialized root cells will pick up water from the soil matrix. It'll flow through the stem of the plant, up the xylem to the leaf, and then be released through the stomata. So you can kind of think of plants as giant living straws that are transporting water from the soil to the atmosphere. And that transport is critical to maintaining their primary metabolism and their hydrostatic pressure. We've all seen plants that haven't been watered, they'll wilt and their physiology won't be at its peak. Next slide. So plants need water, that's unescapable. So we need to find solutions to sustainably manage our water and make our agricultural system more resilient. While not an exhaustive list, I highlighted 3 potential interesting practices: making smarter irrigation choices and precision water management, improved plant breeding for drought and water limitations, and incorporation of the plant and soil microbiota. Next slide. And so what is precision water management? Precision water management entails the implementation of advanced irrigation technologies, effective management of drainage, and utilizing precision water technologies to optimize our water to the field. And in my limited literature review, two major research focus areas emerged: methods to improve irrigation engineering and enhancements to water monitoring in the fields. So irrigation engineering involves improving water delivery to the field through the study of the flow rates, methods, and assessing the trade-offs related to the cost and sustainability of actually moving and transporting water to the field, while monitoring focuses on developing sensors that capture the spatial and temporal variability in agriculture to deliver water to the right place in the right time. And these efforts are crucially important for optimizing our water usage in agriculture and ensuring that the sustainability of our water to face the demands and the environmental stresses plants are feeling in those fields. Next slide. Another approach is to improve the germplasm and actually change the plants to be more productive in our agricultural system. So breeding and selecting for traits that improve the water use efficiency of the plant. And usually this involves for selecting traits at the major water acquisition and water loss regions of the plants. Here I highlighted two examples, papers that I thought were really interesting. The first paper was a plant physiology paper that showed that the stomatal patterning at the leaf surface played a huge role in shaping the water use efficiency of the plant. So basically, with the stomata, the number, the percentage, and the size of the stomata on the leaf surface altered how much water the plant lost during photosynthesis. The second paper that I'd like to highlight showed that roof morphology and root architecture were really important in the uptake of water from the soil matrix and improved the plant's response to these limited water conditions. However, this work is challenging and is difficult, as Jan illustrated. Multiple genes and multiple alleles are involved in this process. There are complex traits that are underlying this, and they're context dependent. So understanding how these things are changing across environment and respond to climate change is instrumental in the future. Next slide. And of course, my favorite solution, and I promise I'm not biased, for reducing our agricultural water usage stems from the incorporation and advancements in plant microbiome sciences. Specifically, we're aiming to harness the plant and soil microbiome within the agricultural system. So we've learned in recent years that plants can alter their microbiome recruitment in response to water limitation, and that this altered microbiome recruitment and microbiome play a role in regulating plants' response to drought and water limitations via hormone production and increased water absorption. So plants have this complex relationship with soil and foliar microbial communities that changes their physiology under these drought stressors. And here I just wanted to highlight some research going on in my lab right now, where we inoculated Setaria flowers with a microbial community from an Arizona soil, so a microbial community that's arid and land adapted, and then inoculated them with a PBS control, and found that in the subsequent generation, when we grew these plants under water stress, those plants that were grown with the Arizona soil inoculum had no yield loss under our drought conditions. So we're basically finding that the soil microbial community buffers the stress and pressures that the plant faces. And we're furthermore exploring the yield Furthermore, we're exploring the mechanisms by which complex microbial communities can change the hydroecology of the rhizosphere to improve plant sustainability and drought response. Next slide. But ultimately, to tackle the challenges of plant health and water management amidst a changing climate, we must integrate strategies from diverse fields of research, leveraging across disciplines. So microbiologists, engineers, plant breeders, climate scientists, and hydrologists all need to come together and cross-pollinate their ideas and come up with the next generation of water management so we can have a long, sustained agricultural system. Next slide. And while resolving our water consumption challenge is paramount in the United States, prioritizing the development of agricultural resistance to climate change will yield benefits for farmers not only nationally, but also on a global scale. As I mentioned previously, irrigation is a global practice with many countries across the globe using irrigation as a reliable food source. So if we make advances here, we'll have advancements across the globe. Thank you so much for your question. Thank you so much for listening, and I'm excited to participate in the discussion later.
Awesome. Well, thank you, Alonso. Fascinating and very near and dear to my heart as a University of Arizona alum myself. On deck, we have Dr. Sharifa Crandall. with our next topic discussing soil as an influence of plant health. Sharifa is the Assistant Professor of Soil-Borne Disease Ecology at the Pennsylvania State University. She studies how and why fungal pathogens drive plant disease dynamics over time and space. Her lab researches soil pathogen biodiversity and function. She also uses microbiome tools to explore how communities of soil microbes can be used to diagnose plant health in specialty crops like potatoes and grapes. Ultimately, Dr. Crandall would like to translate her findings into useful solutions for farmers so they may effectively manage crop diseases. In her free time, she also enjoys hunting for mushrooms. Sharifa, take us away.
Great. Well, thank you so much. And thanks so much for the opportunity to speak today. So I wanted to tell the audience that I was given the directions to really think broad and to really give a general overview about soil health and plant health. So we'll start a little bit with sort of the history of soils through time, and then get into some of the nitty gritty, zoom in, talk a little bit about what are soils, why does it matter, and then zoom out again and talk a little bit about my research and what I do at the local and global level. So next slide. So from rock to soil. So of course the earth is 4.5 billion years old, but it turns out that soils were actually formed around 400 to 450 million years ago. So that's not very long ago. And today they're still being formed by processes of weathering, as well as from decomposition from organisms. And next slide. And so when it comes to ancient agricultural soils, we're, zooming forward here, the Nile River Valley is a great example of where we had agriculture actually occur on a massive scale for the first time in the history of the earth. And so at least that's what we found through records and whatnot. And it turns out it was because there was land that in the Nile Valley that actually was flooded, and then in times of less water and less precipitation, these lands actually the water receded, and this happened over time, and basically you just got really fertile soil. Next slide. So it turns out that another reason that this ancient culture actually was able to en masse work the soil is because they actually used and domesticated animals. And they're not the only culture that did, but it's a great example. Here's a painting from the burial chamber of Sennacherib. It's from 1200 BC, which is fascinating. And you can see they used cattle. Aren't those beautiful cattle? I think they're very handsome. Next slide. And so if you were to actually then move to the Americas, from ancient times to today, there is a lot of agriculture going on, and it's sort of the center of diversity of a lot of vegetable crops, it turns out, like potato, for example. So next slide. And so present day agriculture in the world happens to be, a lot of it happens to be sustainable, subsistence agriculture. And we don't often think about that, maybe if you're from the US, for example, but this is true worldwide. And, but there's also more agriculture that occurs maybe in the US where it's en masse. It happens, for example, there's a Pennsylvania cornfield where you can have monocultures perhaps. And we are moving towards more sustainable solutions, and I'll talk about that a little bit later. But here's an example of a high tunnel polyculture example, again here in Pennsylvania, where you can have different crops growing in the soil, and this can be very useful. So next slide. So present day agriculture, like Dr. Favela actually talked about, does involve a lot of precision agriculture when the resources are available. And this can use a lot of technology. Some people even use drones with remote sensing to monitor maybe plant diseases from the air, as well as patches of maybe drought or other issues. Next slide. And so we do know this, and I think those of you that are gardeners or farmers know this very well, that crop health is intimately tied to soil health. Next slide. So, again, this is a very general talk where we're talking a little bit about hey, what the heck is soil? So it turns out that dirt is not soil, and soil is what scientists actually use, and there's a recipe for soil, and it has 5 ingredients. So minerals are really important. These are like quartz, SiO2, for example, and also soil organic matter. And this happens to be, if you look at the picture on the right, you see that nice brown layer. That's where all the decomposed carbon material is, and it can be very helpful and very useful for crops to grow because that's where there are a lot of nutrients. Soils also predominantly have gas pockets, and they're very porous. So there's air in there as well as water. And so we've talked a little bit about the importance of water and what happens when there isn't water in the soil. This can have high impacts on the plant, the growth and sustenance of many plants worldwide. And of course, there are living organisms in the soil. So here you can actually see in this little pie chart at the bottom that for the components of a silty loam soil, most of it actually happens to be water for this particular example. And then there are a lot of gases again in the pores. So what happens when you have drought-like or abiotic stresses? Well, you lose that water, and sometimes you can even get compaction where those pores become smaller and it's just harder for plants to grow. Next slide. So soil scientists like to talk about soil horizons, and these are really important in terms of thinking about sort of layers and where you might want to actually grow your crops and what the bedrock is actually like underneath and how deep you should be planting, etc. Next slide. Next slide, please. Yes. So there are just different types of soil horizons worldwide, and they're just, it's wonderful. I just wanted to show some different slides here. There are different minerals and different mineralization processes that occur, like the accumulation of red iron oxide. You may have seen soils that are really red, or pyrite-rich soils, which end up having layers of gray and black rock and soil sort of compacted down. Next slide. Okay, so we've talked a little bit about microbiomes. I'm not going to get into gory detail, but we do know, just like human beings who have, we have biotic little organisms, like fungi and bacteria in our gut. So do plants, and they're essential for plant growth and survival. And you can actually isolate some of these. And most of the plants on the planet have these endophytes, endo meaning inside, phyte meaning plant. And you can isolate them and learn a little bit about their roles, their ecological roles. Some of you may have heard of mycorrhizae. These are some great examples underground of fungi that help sequester water from the soil and help with plant growth. And then there are also bacteria. And then most of the time, we just don't know what the role is, the ecological role. So this is something that there's a lot of future work, of future research that could really ask these questions. Next slide. And so a little bit about my own research. I work on Fusarium solani, which is actually a plant pathogen. So it moves through the soil and attacks plant roots. And you can see here, there's a lot of wilting that happens in a field. And if you look on the right, you can see just some really sad soybeans. So it attacks mainly soybeans, but it turns out it's also a soil-borne pathogen on that can cross over and also attack humans, and especially immunocompromised people. And so, some of my work involves looking at this and looking at the phylogenies of these different groups of pathogens. Next slide. Okay, so we've talked a little bit about biotic and abiotic factors, so I'm not going to go into detail. Next slide. Okay, so there are these abiotic and biotic factors that can really affect soil health and plant health. And of course, drought is one of them. And the synergy of these two, as Dr. Leach was talking about, are really important, that synergy between the abiotic and biotic factors in terms of affecting how crops grow. Next slide. And so I just wanted to talk about this general sort of poster child example of the biotic side of soil pathogens, where the Great Potato Famine in Ireland was actually caused by Phytophthora infestans. And this is an oomycete pathogen that basically attacks the potato and caused huge problems with food security, as we know. Next slide. And today we're trying to manage pathogens. This Late blight has not gone away. Phytophthora infestans is still around today, but there are so many other soil-borne pathogens that just need to be studied. And my work really works on, my work is really about looking at researching sustainable solutions for managing these soil-borne pathogens. And so some examples here are listed on the left, such as using different crop covers. You can use legumes, for example, that help increase the nitrogen in your soil, and you can use these in different periods. You can use buffer strips, as well as crop rotations, and crop rotations in particular can really help with suppressing pathogens. Next slide. And so a promising approach that we're actually researching is soil steaming disinfection, where you can actually get rid of the microbiome in the soil and the pathogens, and then look at soil recovery over time. One of the things that we found out is that soils actually recover in nurseries at least 2 months afterwards and kind of look like the microbiome looks like what it was before. Next slide. We do this a lot in strawberries. Next slide. We're almost at the end. We're actually pioneering some work out in California where we're trying to scale up from the nursery level and try to do some more steaming in the field. Next slide. And so now I just wanted to just end for like 30 seconds and just talk a little bit about climate and land use change and this idea of desertification. So now we're zooming out again. And so a lot of my work involves looking at the agricultural region in North Africa called the Sahel, which is actually, because of land use change and climate change, really drying out and becoming desert land. Next slide. And so one of the issues there are these huge dust storms that come through and are problems for human health, but also can carry bioaerosols, which happen to be, they could be pathogens, they could be beneficials, but they're from the soil. Next slide. Okay, so we'll look at global dust storms. They're more frequent, the higher duration over time. And here's some papers highlighting that. Next slide. And so if you think about it, there's 6 million tons of elephants equivalent of dust moving across from Africa to the Americas. Next slide. And so we're building a global surveillance system for that. Next slide. And this is with NASA. And so these are some of the resources available. I guess we didn't have a chance to look at the video, but that's okay. And I just wanted to say that, next slide. So you got to love your soils. You got to learn and share knowledge about them, and you have to be okay with getting your hands dirty. And so I just wanted to thank FFAR, but also my lab group. And so with that, I'm done. Thanks.
Thank you, Sharifa. Fantastic. Yeah, that's fascinating. I think dust is something we can all appreciate, especially as it gets drier and warmer. Lastly, I'd like to introduce Dr. Lisa Offringa. Lisa will bring us home and speak to the last topic, plant health connections, human and global health. Lisa is formally trained medical and nutritional ethnobotanist with a PhD in plant sciences. She has more than 20 years of experience in elucidating the intricate connections between people and plants for the benefit of human health and nutrition. Her education and experience span fieldwork to product development, providing a deep understanding of natural products from farm to bottle. Currently, she is the senior manager of plant biology at Brightseed, a biotechnology company that harnesses the power of artificial intelligence to discover novel bioactives in plants. Lisa, we're all ears.
Thank you so much, Ryan. Good morning. I am so pleased to be here today and speak to you about plant health connections and their effect on human and global health. Next slide. Today, I'm going to discuss plant bioactives in the context of human and global health. Next slide. So global health is suffering and mirroring the health of a suffering planet. As climate change, as we discussed, and dramatic increases in populations challenge our planet, humans are challenged with a society that's promoting the standard American diet and pushing packaged and processed foods. The result is shown in exhaustive studies like the Global Burden of Disease Study, which is the world's most comprehensive analysis of how diet impacts human health. The study shows that dramatically under-consuming whole vegetables, fruits, nuts, and grains, was found to be 3 times more deadly than over-consuming the bad factors of things like sugar, salt, and fat. Still, over 90% of Americans fall short of the daily intake of recommendations for vegetables and fruits. Vegetables and fruits provide macronutrients like fiber. They also include micronutrients like vitamins and minerals. They also contain small molecule bioactives. And these small bioactive molecules have been found to increase health and well-being. Next slide. Thank you. So what are bioactives? There are a few definitions put forth by a couple of different institutions. And so we're going to go through those quickly and then find the commonalities. The NIH Office of Dietary Supplements clarifies that bioactives are constituents, but they're not needed to meet the basic human nutritional needs. The National Cancer Institute puts forth that they may promote good health. And the American Dietetics Association explains that they can be found in foods or in dietary supplements. But there's one thing that they all have in common in their similar message is that these are physiological active constituents, and they are all primarily found in food. Next slide, please. So then where do bioactives come from? I think as we've discussed today so far, plants are miraculous creations. Each leaf can adapt to its environment by shifting directions toward the sunlight, and plants literally create themselves. Regardless of how many times I've studied photosynthesis in my career, this continues to blow my mind. They're also masters of adaptation, which is why they create secondary compounds. Every action of a plant is meticulously designed to improve the probability of the survival of that specific unique species. They cannot move, so they have to create these compounds to protect themselves. So plants create these compounds that impact human biology, and yet these small molecules are the unsung heroes of our health. They activate pathways in the human body to benefit health and wellness, and these bioactives are created by plants for their benefit, but they can also benefit the health of humans. For example, a very large portion of the population ingests some form of caffeine. I am on the West Coast, so I definitely ingested some caffeine this morning. So for a plant being munched on by a hungry insect, it adapts by creating caffeine, which is unpalatable to the insect, but it's very helpful to other organisms like humans, unlike me today. Next slide, please. Bioactives are found all around us. They're found in our food, in our medicines, in our spices. So they're found in medicinal plants like the willow tree, whose bark provides us with aspirin. They're found in the French lilac for metformin. And they're also found in common foods like lycopene in tomatoes and lutein in leafy greens. And then finally, the powerhouse curcumin is found in the spice turmeric, which was originally used extensively in Indian and South Asian cooking. These bioactives are prevalent in the plant kingdom, and they offer a rich opportunity to maintain and optimize health on a daily basis. So consuming these bioactives in your food is the basis of food as medicine, in that eating whole foods is good for your body, and it's also good for the planet. Next slide. Thank you. So now we understand why plants form secondary metabolites that can be bioactive, and some examples of these bioactives as useful compounds for human health. The secondary metabolites that are in nature, an only tiny fraction of them are known and annotated. The ones that we do know about, we've been able to use for therapeutic purposes by either using them as natural products or as natural products derivatives or analogs of them, which is how 60% of clinical drugs came into being. So if we could accomplish this much with what little we know today, imagine how much we can change the face of human health when we tap into more of these elusive plant compounds. Next slide, please. At Brightseed, we are actively looking for the bioactives in nature using machine learning and multiomics to unlock the restorative power of nature for human health. These bioactives work in the body like a key fits into a lock, unlocking health benefits at the molecular level. At the core of our work is AI, which is a great tool to rapidly look for all the keys in plants to unlock all the locks in the human body. And so from this, we know nature down to the molecular level, giving us an unparalleled insight into plants, bioactives, and their impact on human health. Next slide, please. We consider these unknown and unannotated bioactives in nature as the dark matter, and the vast majority are still hidden in nature and yet to be explored. So many of these opportunities are yet to be discovered in the dark matter, and we're looking to shed light on our understanding on how plants affect the body in a profound way. Next slide, please. So Forager AI is at the core of what we do at Brightseed. Forager utilizes machine learning and our proprietary data sets shown here to predict bioactives from the 7 million plant compounds we've already mapped. This is 70 times more than what's known in published scientific literature, with more than 40,000 predicted bioactives over 23 health territories. These databases have grown over time to power Forager, and through machine learning, these predictions strengthen. Forager offers a faster and more targeted path to discovery and de-risks the process of companies seeking to innovate new health products. We can start with the health benefit and leverage our massive data set of plant compounds to find bioactives that align to biological receptors in the body associated with that specific health benefit. Built on our proprietary machine learning technology, Forager works in 3 parts. First, it catalogs small molecule bioactives in the plant kingdom. It predicts the health benefits provided by these bioactives for the body. And then it also knows which plants or natural compounds contain each bioactive compound. We also are able to identify novel bioactives uniquely created in the gut microbiome from plant and bacteria interactions. Next slide, please. So using AI, we can also look to the different opportunities to identify and in fact even boost bioactives in our food. It's well known, as Dr. Crandall stated, that nutrient content of food is increased when soil quality is excellent. Through sustainable agriculture and farming, we can guide farmers on which crops to grow by evaluating compounds and potential bioactives in them to increase their healthfulness. Through supply chains and productions, AI can optimize foods to enhance their bioactive compounds within the supply chain and therefore increase food quality. And then finally, through consumer goods, whole foods can be characterized by their bioactives, and dietary supplements can be created to provide consumers with more ways to maintain their health and well-being. So imagine how incredible it would be if you looked at your nutrition facts label and saw your vitamins, your minerals, and your bioactives listed there. Next slide, please. Additionally, AI can contribute to planetary health. For example, it can help in optimizing plant cultivation in cell culture platforms by selecting the best ways to initiate different plant growth. This can help encourage greater crop variety in farming while maximizing the desired characteristics like bioactives in plants. We can identify side streams or waste streams that may contain bioactives to monetize them for companies and reduce waste. And AI can also help to guide farming practices to optimize water and fertilizer usage by accounting for weather and climate change that are affecting the yield of different crops. AI can be used in these many different ways to address issues that face our planet in a way that increases human and global health. Next slide, please. So as bioactives are discovered in research, it emphasizes the need for nature, for its preservation, and for the sustainable use of its resources. Without nature, we lose so much richness in our lives and opportunities to optimize our health and the health of the planet. So I want to thank you again for having me today. Thank you so much, Ryan and FFAR, and congratulations again on your 10-year anniversary. Thank you so much.
Well, thank you, Lisa. That was fascinating, and I need to eat more turmeric. Thank you for the reminder. Thank you to all of our panelists. I really appreciate your time today and your willingness to share your passion and your expertise. With that, I would like to open the floor now to our Q&A segment. My colleague Ryan Conley will MC and kind of read the questions out loud in the chat to our panelists to respond. Ryan, what do we have?
Hi, good morning. So our first comment is not quite a question, it's a comment for Dr. Leach. Thank you for your insights. Your idea of integrating system approach and breeding to mitigate diseases and pathogen is realistic. I believe understanding the neural network and cell signaling at the cell level is crucial. The next question, I'm sorry, did you?
I was just going to say, I totally agree.
My next question, I believe, is for Dr. Crandall. what are the key indicators of soil health that you assess in evaluating the overall health and productivity of a soil system?
Oh, that's a fantastic question. So there are, in general, different, I would say maybe 4 different things you can think about. And the first would be sort of the physical components, the chemical, the biological, and within, the fourth would be probably soil organic matter. So first, organic matter is really important. That actually helps you understand how much nutrient availability there is in the soil. Other sort of important things for the physical piece would be looking at soil depth, for example, and soil texture. And soil texture helps you understand, we talked about silt and other types of minerals, for example. I'll tell you more about the water holding capacity. So that's a very important sort of component as well. And then finally, for biological, if you don't have a microscope, for example, hopefully, folks do, but if you don't and you can't see those little organisms in the soil, like mycorrhizae, for example, sometimes you can see them on the roots, you can see the mycelium. And so that's a good indicator that maybe your plant is, or your crop is getting the nutrients it really needs through the symbiotic relationship. Other things you can look at are earthworms. But then again, there are these invasive earthworms in different parts of the world, so you have to be a little careful with that. But these are some indicators. So I hope that answered your question.
Right. Thank you.
Thank you. We have one more. So if anyone has any questions, be sure to, you can drop them in the chat or the Q&A. This is the last one we have for now. For Dr. Offringa, Lisa, wonderful presentation, and I love the AI approach to unravel and discover bioactives. Not sure if you touched on how the approach can be applied for identifying new bioactives for plant protection and green biology.
Sure, thanks. This is a great question. And I didn't touch on this. We focus primarily on bioactives for human health. So not really looking so much at plant protection and green biology. That being said, I think thinking about plant diversity and thinking about how to find the most sustainable plant source for bioactives. One of the things that Forager is able to do that I think is so amazing is, once we've identified a bioactive, Forager is able to look back into its database and find different plants that also contain that bioactive or predicted to have that bioactive. And so for that reason, if you were looking at some kind of plant protection, or if you were, looking at trying to find a more sustainable plant for one that you found a useful bioactive in, you could feed that information back into Forager, and Forager could come out with a list of different plants where that bioactive may be present.
Well,
Thank you. Next question, I believe, is open for anyone. What are some cost-effective and sustainable methods for maintaining and improving crop health and productivity in a large-scale agricultural operation?
I can try to start with that. Maybe other folks can. Sorry, Jan, I think you wanted.
No, go ahead.
Okay, well, I was thinking too, just off the top of my head, would be crop rotations. If you have different crops that you normally rotate in during the growing season or between growing seasons, that's great. For example, here in California, in the US, broccoli and lettuce are rotated. And the reason for that is to suppress diseases. So that improves crop health. So, broccoli it might not be a host for certain pathogens in the soil. But lettuce does have that susceptibility, but then you can actually rotate them out. So one year the pathogen doesn't have its host. So the pathogen doesn't really do anything, and you can have an increase in yield if you rotate. And the second would be cover crops. If you have any legumes that you can use, in between seasons, for example, when you're growing, legumes add nitrogen to the soil. So that's another way. Go ahead, Jan.
I would have touched on the same things. I would also say paying attention to the genetics of the materials that you're planting and choosing those materials wisely in terms of disease resistance, the use of water is important in different areas of the world, how much water you're using. So, of course, these are pretty standard practices, but sometimes I think we forget these integrated approaches when we think about how to do things.
And if I could just
Oops.
chime in to answer this a little bit, to highlight some of the other work I've done. So I've worked a lot on biological nitrification inhibition and how some genetic cultivars can suppress these nitrifiers. So I think looking for the next sort of generation of crops that have these characteristics that interact with microbial communities that allow us to put less inputs, less fertilizers, and overall into our agricultural fields will result in huge gains in cost effectiveness because we won't have to buy all those inputs and just have it in the seed and in the genotypes ready to go.
Ryan, I think we have maybe time for one more, and then we should wrap up here shortly.
Yeah, one more. And if you do have any other questions, you can email Ryan Comella at rcomella@foundationfar.org. I'm volunteering you to do this. We can pass it on to our panelists and get responses back because we do have about a few more, but I'll read the most in order. The final one will be, are there any ways to help alleviate the effects of the intensifying sandstorms carrying sand from the Sahara to Europe? Is there much research on the topic?
Hey, I love this question. Number one, there isn't much research, although certain countries are trying to deal with this problem. So there are also dust storms from China, for example, especially in those areas that used to be more agricultural or forested. And so the Chinese government is trying to do what we call afforestation, which is actually grow trees where there's a lot of desert or desert is sort of encroaching upon agricultural land. Whether that's working or not, that's very difficult. They're trying to grow like millions of trees because trees kind of hold the dust and, the soil down. But that's one technique. But really, there isn't much out there. These are large-scale climate phenomenons. They happen annually across the world, and they carry a bunch of not just dust particulates, but also bioaerosols and potentially and definitely microbes. But we're still trying to figure out what those microbes are and their viability in the air as well. So my research program does that. But thanks for the question.
Okay. Wow. Yeah. Thank you. I think that will conclude our session today. As Ryan mentioned, for any questions or inquiries, you can find our contact information on the screen here. I'd just like to take a second to personally thank our panelists for lending your time and expertise to help make this webinar really shine. Thank you all for your passion and sharing that with us today. And thank you to our attendees for your engagement and lending us your ears on a Tuesday. With that, happy Tuesday, everyone, and thank you for celebrating an International Day of Plant Health and our decadal anniversary. Thanks all.
Thanks so much, Ryan.
Thanks. Bye.
Bye, everyone.